Okay, let's get started. Very good. Excellent. Okay, so today we will continue to talk about the diffusion and very important, very relevant experimental techniques. How can we categorize diffusion? Especially how can we measure the diffusion coefficient, capital D, right? So before that, a few logistics updates and some clarification. First of all, the homeworks, you should have all the homeworks solution now, number one, number two. And then homework number 3 is uploaded. So you can work on that now. The question there is associated to the experimental techniques we are going to discuss. And then the clarification I want to give is about the midterm exam number 2. So in the midterm exam number 2, you will do basically 2 things. Can you hear me? Can you hear me? Yeah.
Wait.
Good. First thing is that you will present your project, mostly the idea and the plans you are going to do for the final projects. That will take about 10 to 12 minutes. And then we will leave 2 to 3 minutes for your audience, for your peers to ask you questions, and then for you to answer that. Okay, so that's the presentation. The score of your exam number 2 is determined by that. And then second thing is, after the presentation, you can send me your writing proposal. So that actually you can prepare now. This writing proposal, I showed the format in previous lectures, right? So now you can prepare a writing proposal. However, this writing proposal mainly is for you to, for me to give you the comments. So based on your proposal, you write it as careful as possible, as complete as possible. Then I will give you the comments that will help you to revise that in the time to come. Okay, then that will help you. Then you can, after your revision, you gradually develop it as you develop your project for the final project. Then this proposal will become the final report you can send to me. Okay, so this is a chance I can help you to revise your writing draft. Okay, so you can send after the presentation, your score is determined, but you can send, and then you can send me your proposal and also your PPT. Okay, so then I will comment the proposal and then send it back to you for you to improve and upgrade that into the final report. Okay, so these are 2 things you will do for the midterm exam. It's coming. It's end of the October, right?
I have a quick question. So the writing proposal, is that due the same day that we present, or is it due the first day of presentations?
After the presentation is fine. Yeah.
Okay.
Yeah.
Thank you.
After presentation, within 1 week should be okay. Yeah.
Okay. Thank you.
Good. Very good. Yeah. Any other questions? I have some excellent examples in the Canvas already, so you can use that to as a guidance for you to learn how to write well in terms of the proposal and the final report. Okay. And then
I've got a quick question.
Yeah, please.
So in general, the presentation is supposed to introduce the general subject of your topic and the science surrounding it, the literature surrounding it, and then go into what you specifically want to study, like your specific question that you're designing research around or a research project around.
Yes.
Okay, and then the written proposal is just that same concept but more developed.
Concept, but in a writing format, and remember last time when I show you the format of writing, you first talk about the introduction, motivation, and then significance, and then have the specific aims, different specific aims. That aims are exactly the things you propose you will do for your final project.
Okay, I gotcha. Very
good questions. Yeah, very good. So all of you can start to finalize your teammates, okay, your partners, and then email me your name, the team members' name, and then I can determine how many teams all together we will have in our class. Then I can schedule a time for you. Okay, so everybody, I will allocate 15 minutes for you. Okay, good, very good. Okay, that's all for the midterm exam number 2. Very important, and it's a very good practice opportunity for you to learn how to determine a project, how to present a project, and how to convert the presentation into a formal proposal format. Okay, and then how to improve your proposal based on my comments and based on the questions raised by your peers, okay, in the class. Okay. And then you can develop the final project. And I mentioned at the very beginning of the semester that the final project should contain original idea, original thoughts, modeling, or the proposed new experiment techniques, right? So, or develop new experiment techniques. So those should be presented very well in your midterm exam number 2 and also the final exam. Okay, so this actually is a very good research opportunity for you. Okay, and let me know anything I can help you with. Okay, can you guys hear me?
Yep,
yeah,
we hear you.
Very good, very good. Okay, that's all about the midterm exam number 2. And then, yeah, I already the rest of the agendas I have explained in the last lecture. We will have 2 invited speakers, 2 distinguished invited speakers coming in the same format in the Zoom with us. By the way, how about how do you feel about the presentation, the research presentation given by our invited speaker, Professor Brent Reynolds? What's your feedback?
I thought it was really interesting.
Good, good. Yeah, that's a very important area. So, and Professor Reynolds is an excellent scientist. He has been doing great science in the neural stem cell for several decades. So he's really the expert in that area. Okay. Any other thoughts?
I really liked how he connected his research to like day-to-day life, and showed how his research impacts us in the day-to-day.
Good.
I think that was important to highlight.
Yeah, very good, very good. Thank you, Daniel. Good. Yeah, he likes to talk with all of us. So he said the students really ask great questions. So you guys also did a good job. Okay, good. So in the future, we have 4 invited lecture actually. In the end of the November and the beginning of December, we also have another 2. So those are all very good scientists, and they will give a talk. And you make sure that you learn well. I hope all of you learn well and ask great questions, bold and great questions. Okay. Okay. So, and feel free to submit the survey through the email to me if you have any things that you think can further improve your learning experience. Okay. So I'll be very happy to help you. Okay. Very good. So that's all for the logistics. If there are no more questions, then let's continue our class and focus on our science here. Good. So right now we are here. We're in the diffusion. We have finished the probability theory, Brownian motion. We have studied the diffusion. We talked about the correlation now. So now we will continue to talk about its applications and more technologies used to measure the diffusion. Okay. After that, we will talk about the important optics imaging and optical method used in the biophysics. Okay. And that actually is associated with the membrane potential ion channel. Because we can use the optical imaging to measure membrane potential and then derive the dynamics of ion channel and the proteins, their dynamics on the cell membranes. Okay, so those are associated together, as well as the intermolecular force. Force can also be imaged by the optical method. Okay. Good. So we have in last lecture we have talked about the diffusion equation, and we used the differentiation method to describe the diffusion. So we we derived the diffusion equation using the following method, right? So we divide the whole volume into many thin slices, and then denote the concentrations of all the particles in different slices as C0x, and before they diffuse, before they move. And then after they move, they will follow the diffusion equation, and also by quantify the transport of particles purely through any interface between two slides. We carefully drafted this expression, right? J is the flux. So we carefully drafted the quantitative expression for the J. Okay, this is the J has the unit number of particle per area. Actually, also is per time. Okay, number of particles pass through the interface per unit area per time per second. Okay, so then this J can be written in this way, and then this equation we can input the diffusion equation input it in, and then find out this J can be written as minus diffusion coefficient D, capital D, delta x squared divided by 2 delta t, right? So this is a one-dimensional condition, so it's 2 delta t. Then this is capital D, and then have another term, rest term. This is simply the gradient, spatial gradient of the concentration. Okay, so that concentration is the current concentration. Okay, it's not at the beginning, because it keeps changing. So C is a function of the x, and J is expressed as the minus J equals minus D gradient C. If it's in 1D, then it's a partial C, partial X. But in 2D or 3D, you can write it as a delta, inverse delta. That's called gradient C, right? That's J, the flux. Then we also, and that is called Fick's law. And after that, we use the divergence theory in the mathematics. Okay, divergence theory. So that and also the mass conservation theory. Based on the mass conservation, for any enclosed volume, the change of the mass inside the volume is partial C, partial T, right? It's changing partial C, partial T. Here, actually, you can do an integration with the volume, then get the total mass inside. But if you only consider the concentration change inside, then it's partial C, partial T. And then this concentration change is due to the sum of the incoming flux and output in flux. Okay, so in and out combined together, pure transport of the particles through the surface of this volume, right? That based on the convergence theory, that is the minus delta dot J, right? Because right, minus means we consider it in as the positive sign, positive direction. So then it's a minus delta dot J as convergence theory. Then based on this, we also know J equals to minus D gradient C. Then this putting together gives this very important equation, partial C, partial T, temporal change of concentration equals to D times delta square C. Okay, so this is the spatial change of the concentration. Okay, spatial change of concentration gave rise to the flow of the J, flow of the flux. That equals to that dot product gradient equals to the temporal change of the concentration. Okay? So this equation is very useful in the physics, mathematical physics. Okay, so the name is called the diffusion equation because it's originally derived in the way we just derived, based on the diffusion theory. Okay, but this equation format, can be it can be found, this format can be found in many other physics topics. So if other physics topics use the same kind of equation, even the variable are different, then the change, the physical property characteristic change is similar. So if you understand the one condition here, other condition it will be very similar. You can derive that in the same way. Okay. So this is an important equation called diffusion equation. Now, this equation, if the barrier, so in this equation, if we want to calculate this equation, we want to understand we want to give the solution of this equation, then we need to put the boundary condition, right? We put the boundary condition and then solve this equation, because this is the differential equation. When you do the integration, you reverse it, integrate it, then you will have the constant. Then for the constant to solve them, to know their exact value, you need to combine with the boundary conditions, right? So in the years, for years that people, yeah, Cole, you have a question, please go ahead.
Yeah, sorry, I wanted to ask, just to clarify on the diffusion equation. So this is saying that the change in concentration, or the temporal rate of change of concentration, is dependent on C, the current instantaneous concentration in that space?
Yes.
Okay, and
Because
so
yeah, go ahead.
okay, and the proportionality is d times the gradient squared. I'm a bit confused by the gradient squared, because I know that to be a vector value. So I'm not really sure how that works here.
Good. Let me explain it. Yeah. So the gradient, you see my drawing? Gradient.
Yep, I can see it.
So gradient, usually this term usually is a gradient of any value. This value usually is the scalar. Okay, it's a value, scalar. It's not a vector. C is a scalar. It's a value. Okay, no direction.
Right.
Now, if you take the gradient of it, then it actually gives you the direction that this concentration change most. Okay, that is a gradient, right? So that's actually is partial C, partial X times unit vector I. So this is a vector. Okay, and then another one is partial C. In the future, I will put it in the slides. Partial C, partial Y. J. That is another unit vector around the Y direction. Y axis.
Right.
And then the last one is similar, partial C, partial Z, in terms of the K, IJK. So this tells us the change of the concentration around three directions. And then depends on their value in each direction. Then these three together forms a new vector, forms a vector. That is this delta C, right? This is delta C. So that tells us where the concentration moves the fast, changes the most dramatically. Okay.
Okay, interesting. But if C itself represents a single scalar, so in other words, it's representing the instantaneous concentration at only a single point,
Yeah.
then isn't the derivative of a scalar just zero?
It's C is not a single point in one point, in one dot. C is actually C is a function of X, Y, Z.
That's what, okay, that's what was getting me tripped up. I forgot that C is our concentration function. In that case, okay, that makes perfect sense now.
Yeah. And then we show the divergence theory here. Delta. So this delta actually, the gradient is actually the gradient actually is this guy. Is this guy.
Right, the gradient is ddxi hat plus ddyj hat plus ddzk hat.
Exactly, but they all contain ijk unit vector. Okay, and
Right.
then so gradient symbol by itself is also a vector. Okay, and then that symbol, that vector dot product delta c together, then it goes back to a scalar, right? So then you have a partial c square plus partial c square partial x square plus partial c square partial y square, right?
Right.
So that is this one. Partial c.
Okay. And in this case, the effect of squaring it, I suppose like anything, squaring it removes any negative values. So do we just square it to get a better idea of magnitude, or why exactly are we squaring it?
Here this square is not the that square. It's actually to take 2 times derivative. It can be negative, right? So this 2 times
Right.
derivative. Yeah.
Okay. Okay, that's making more sense now.
Very good. Very good. Yeah. Anybody ask questions whenever you have. Okay. So then I can clarify it in situ. Can everybody hear me?
Yeah.
Yeah. Okay.
Yeah.
Okay. Good. Good. So that's the expression for the diffusion equation that we expanded it. Okay. So this term equals to delta square c. Yeah. Good. Good. Okay. Good. Now let's go back. So we have this recorded, so you can see it afterwards. So let me remove it and go to the next stance. Okay, good. So to solve that diffusion equation, as we said, we need to apply the boundary conditions. Right? So those boundary conditions, depending on different physics conditions, actually there are many boundary conditions quite common to encounter in the real life, in a lab. Okay, you always encounter those boundary conditions. There are 2 types of boundary conditions we often encounter. First one is here. It's called impermeable barrier. Impermeable barrier. That means that the surface of the volume I mentioned that is closed for certain element. It's resistant to certain elements, and then those elements cannot go through that surface. You know? So that kind of a surface is called impermeable barrier. So in that case, no flux of particle can go across that surface. Then the change of we can write the boundary condition as the n dot. So this n is actually the vertical, the normal vector on that surface. Normal vector on that surface. So it's a vector, unit vector, perpendicular to the surface. Okay. So that dot product gradient of the c equal to 0. So that element cannot go through the surface. So this happens a lot. For example, if we have the membrane, cell membrane, some of the membrane in the lab we can actually synthesize that. Then those synthesized membrane will not contain any protein on in the membrane. Then the water molecule cannot go through anymore. So all the water inside that volume synthesized, enclosed by membrane, remain inside the volume. That volume is called vesicle, synthesized vesicle. So then the water inside, when the vesicle was generated, stay inside. The water outside stay outside. So there's no exchange of the water. Okay? That's totally different from the real cell. The real cell, they have the water channel on the membrane. So then they can let the water get in. But the real cell cannot let any element get in. Right? Some kind of element cannot get in. Then for that kind of element, the membrane is an impermeable barrier. Okay? So this happens a lot when we, and it's element type specific. For different, when people do the modeling, when different elements, some of them, they will use this boundary condition. Some others use other boundary conditions. Okay? So this is one. Second one is called clamped concentration at the boundary. Means the concentration of that element, concentration on the surface actually is fixed. Okay? So C is fixed. It will not change. Even partial C, partial T is there. So inside the concentration could change, but on the boundary, the concentration is fixed. Okay, and then the flux can be anything. Okay, the flux can be anything. Flux is coming from this C, right? So there are many examples. For example, this, by the way, this is called a clamped concentration. I want to emphasize this word, clamped. Clamped always means it's fixed. Okay, so when you have a chemical clamp, in many cases the concentration is concentration of the chemical on the surface, on the volume, select the volume, their surface is fixed, or many times the gradient is fixed. Okay, gradient is fixed. This all happens. This is all called a chemical clamp. This happens when you have a single cell in a bigger petri dish, and then the concentration of the chemical in a bigger petri dish will not change. Even the cell absorbs some chemical into the cell body, because cell is in a bigger reservoir. So bigger reservoir will not change. So then the chemical is clamped. That's one. Second one is called the electrical clamp. So this one is in the cell membrane, as we said, we have the voltage there, right? And then the gradient change of the voltage usually induce electric current, because the membrane generates an electric field that it can let the charged particles to get into the membrane. And then when the charged particle get in, because it carries the charge, so it actually generates a current, right? So current is like the gradient, right? Gradient delta, gradient C, right? It's like that. It becomes the flux, right? But in the lab, there are some electronic devices that can hold the cell membrane, and then apply a big voltage on the cell membrane, and then let the cell membrane to be constant. Even there are current getting in, but they can hold the membrane current to be constant. That is called a voltage clamp. Voltage clamp. Okay. Alternatively, you can also let that device to constantly inject the current into the cell or out of the cell, no matter what voltage the membrane has at that moment. Okay, that is called a current clamp. Okay, both conditions can be realized in a lab. You can use the tools to realize that. And then once this is realized, then the cell will have really interesting function happens. Okay, and using these tools, using those electrical clamp, you can study the dynamics of a single transmembrane protein in the cell. Okay, if that protein, their conformation change is dependent on the membrane voltage, then you can clamp voltage to like a -70 millivolt, and then suddenly change it to -20 millivolt, and then clamp there. Then you can see in these two different voltage magnitudes how the transmembrane protein change, right? Then people actually, if you want to study the protein conformation, you can use this way to quickly change the voltage, and then apply the cold temperature immediately to let the membrane, to let the cell to be fixed, and then the protein conformation is also fixed there. Then they treat the membrane, purify the membrane, and then take those membrane into the electrical microscope. Then you can see directly in two different conditions what's the shape of the transmembrane proteins. Right? So that's how people find out the different conformation of the proteins. Right now,
Dr. Tang, I have a question.
Yeah, please.
So I guess I'm kind of confused here, where if we're fixing the concentration, then the change in concentration is 0, which means the flux would be 0 as well, right? Or is it concentrations fixed in one space, but there's still a gradient across it, so it's fixed. I guess that's where I'm confused. If the concentration's fixed, how can you have a flux?
Oh, what I'm saying is the concentration is fixed in one side on the boundary. Okay.
Okay, so there's still a gradient. Just one specific point in space is fixed.
You still have a gradient, but the one side of the surface, the concentration will not change. For example, if you have a bacteria will keep eating energy. If you have an ATP molecule there, bacteria keep eating the ATP molecule and then perform the metabolism by the cell, right? If there are many food, many ATP outside in the nature, then bacteria will sit there, keep absorbing the ATP. So the inside of bacteria, ATP is keep absorbed. So and the ATP to keep going through the cell membrane, get into the cell bacteria's the body. But the environment is very rich. There are many ATP there. So if bacteria consume 1 ATP or 20 ATP, doesn't matter, because outside have billions of ATP there, right? So then the concentration outside ATP is constant. You got my point? That is called chemically clamped. Outside will not change, right?
Okay. Yeah, I see.
Yeah, very good. Those are good questions. We actually have some example questions in the next few slides. Then you can apply those boundary conditions to those problems. Okay. Good, Jackson. Very good. So that's the chemical and the electrical clamps. There are also mechanical clamps. So you can also apply the mechanical force to let the force on the membrane to be constant, like a tension to become constant. Okay. And also in some cases, the people actually clamp the strength, clamp the deformation of the cell. For example, you can have a specific geometry let the cell get in, and then cell will fill up that space. Like if I have a pattern, you have a certain pattern there, cell will fill up that pattern. So the strength of the cell will be the constant. Okay, so the deformation is constant. That is the mechanical clamp. Okay, so all those clamps will define the boundary conditions of the diffusion problems, and then that can influence the flux, the dynamics. Mm-hmm. So that's all. That's for the theoretical part of diffusion equation. In reality, people also want to use experiments to measure diffusion coefficient. So mainly there are many new methods that keep getting developed, but there are 3 very conventional methods I listed here. Those are the methods that people use to measure the diffusion coefficient. We will explain them one by one. First one is called single molecule tracking. So this one is this method uses the fundamental understanding of the diffusion. Basically, use the first principle to measure diffusion coefficient. We know that when the particle or molecule diffuse, they will have the random walk, right? So then we can actually use the microscope, use the high-speed camera to take videos of the particles. And then you can see in this picture, it shows different frames of the video, right? So each frame, particles are in one specific location. And then different frame will show them in different location. By connecting the direction, connecting the position of the particle in each frame together, you will form these orange lines. That's the trajectories. You will form these trajectories, right? And then this tracking and this measurement can be automatic. There are many automated software developed to do this kind of imaging analysis and do the calculation. And then based on this trajectory, you can plot the delta x. This is a 2D case, right? 2D case. Delta x square, mean square displacement in the delta x in the x direction, mean square displacement in the y direction, right? And then this mean square displacement multiply together because the x, y direction are independent from each other. So they summing together, they become a function of the time, right? Here, by changing the time, changing different time, you can get a different delta x square, right? So for example, if the time is very small, it's not like one step only one step. It's not necessarily the first step, delta x squared plus delta y squared, not the first step. It can be all the frames. As long as the frame to frame time interval is delta t, then they will give us one value of the mean square displacement. So when the time is very small, you get many those value. When the time is bigger, then you get more, you get less. When the time equals to the final duration of the video, you get only one value, right? So then, based on that, you can connect the dots together, form a curve. Okay, that curve showing the is on the bottom on here. That is the trajectory, the curve that we can use to derive the diffusion coefficient. For this case is 2D, then you get 4D here, right? For the in terms of the parameter. And then if there is a real diffusion, then we will get the green curve is a linear. MSD versus time is linear. Then the slope is 4D. If it's confined less than even less than diffusion. So for example, here it's called anomalous diffusion. So you have the confinement there. The particle cannot go too far away. Then the curve will look different. And then there are many other theory developed to analyze this kind of curve. Okay. There are also other curves that go even faster. That it has some active components associated with them. Right? So this actually is the very conventional way. If you want to measure diffusion coefficient of a particle in certain environment, you use this way. That's most direct. Very not very easy to make mistakes. Okay, but to make this measurement really correct, there are also some cautions we need to keep in mind, so that we avoid possible errors. So this condition, the question to be here in the mind is when we perform those measurements, how to account for the error in our localization measurement. So for this particle, each particle, they still have a certain size in the image, occupy certain pixels. So we should pinpoint the center, the core of that particle. Then how precise is your localized method to pinpoint the center? Sometimes people use the Gaussian fitting to pinpoint the center. Sometimes people use other kind of measurement. But if whatever measurement we use, we need to make sure the center is the pinpoint, and the measurement, this localization measurement, is consistent throughout the whole video, right? And also, what to do if the molecule are confined or running into a boundary. So if they always run into a boundary, such as inside a cell or inside an energy well, right? In many cases, there's an energy well there. For example, there's an electric field there. Then the distribution, the movement of the particle is actually not that random. They prefer to go to the boundary. In that case, even we can get this curve, how can we use a different? How can we rectify it? Use some equations to adjust the final parameter, right? To make it be more precise. The third question is how can we compensate if there's a net flow? In many cases that in the experiment, especially in vivo, there's a blood flow keeps on going. Then that will move the particle in one direction. Okay, and then because the heart is beating, so the flow actually is not going in one way uniform. It go forward and then go backwards when the heart is beating. You absorb contracting, heart is contracting, right? So those cyclical movement actually apply the force to the particle, making those analysis to be more interesting. Okay, so we need to know this and then to analyze them carefully. And also, what to do if we have many particles or many molecules in the petri dish, but they are actually of different molecular weight or different polarity, right? So if the molecule are heterogeneous, then everyone have different diffusion coefficient. In that case, how can we differentiate that? and then measure the correct diffusion coefficient of a specific molecule of interest, right? So those are interesting, very interesting questions practically we will encounter in the lab. And then those questions we should bear in mind to clarify that so that we can get the correct results. Okay. Can you guys hear me?
Yeah, I actually have another question on this.
Please,
How can they restrict movement to two axes? Like what's preventing particles from diffusing out of focus for whatever they're like microscope they're imaging it on? Like they have to have some sort of like microfluidic chamber that prevents them from moving in what would be the Z direction, I guess.
Yeah. So you are talking about the Z direction to autofocus, right? So there are different ways people can do. One way is you can use the transparent glass slide, make a chamber. That chamber in the two transparent glass is very close in the Z direction. So the particle will not go too much on the Z latitude. So they will be confined in a thin layer in the XY plane. Okay. So that's
but that doesn't change their like diffusive behavior in X and Y, it bumping off of the Z.
Yeah, because X, Y, Z, they are independent. Right? They are independent. So they only measure the horizontal plane.
Okay.
Good. Very good.
I also have a question.
Yeah.
So would the molecule leaving the field of view, like going beyond the field of view, affect the the data you're getting at all, or no? Does it have to stay within the certain field of view of the microscope?
That depends on what we are measuring. If the region you are interested in is beyond the microscope field of view, then you actually can change the microscope setup to make the field of view to be even larger, to cover the whole space. Okay, so all the experiment designed is to let the field of view in the focus be the region that we have interest. Okay, if they go out of the field of view, then you need to chase the particle, go to that domain, right?
Okay, that makes sense.
Sure. Good. Those are all very good questions. The practical question. I think it would be great that in the future, in the research, we can discuss those different scenarios further. Okay, those are all very interesting. Yeah, good. So that's one method to calculate the diffusion coefficient. Another method is called FRAP. FRAP is the full name of FRAP is fluorescent recovery after photo bleaching. Okay? So this method also very useful to measure diffusion coefficient, especially diffusion coefficient in a plane, for the molecule in a plane, and they freely move all the time in the plane. Okay, such as a cell membrane. Okay, so as we discussed before, cell membrane within the membrane, the molecules can freely move. In fact, the membrane can also jump, can jump upside down within the membrane, because the membrane can be very fluid. Okay, so the particles are keep moving. Now, the mechanism of FRAP is because those particles are keep moving, and then we want to know the diffusion coefficient of those particles. Then, how about we label those particles or those proteins with a fluorescent dye or fluorescent protein, so that whenever the protein move, the target protein move, we can we can trace that by the fluorescence, right? Once we do that, then you can either measure the fluorescent protein movement within the membrane using the first method, as we showed the single molecule movement, or if those fluorescent protein, if the target protein are too many, they are all over the membrane, then it's very hard to measure single one, right? If that's the case, then people always choose FRAP method. This method actually, the mechanism is we can see this cell. See this cell, they have a all over green color on the membrane, right? Now, once they have those all over there, don't worry about this. Don't worry about this. Okay, those are the small spots that do not contain those fluorescence. I'm saying this part, those rest part. Now, if we want to know the diffusion dynamics of the proteins, those green proteins, one way is to use the light, very strong light, to bleach one part of the cell membrane. Okay, so then the very strong light will bleach, make those fluorescent protein lose all their capability to emit photons anymore. Okay, so then you can see the center part here, close to the nuclear part. This part, there are no fluorescent now because the people use light to concentrate this region. So then they're gone. Once they are gone, it's actually only that once this here become darker, that means fluorescent protein lose the emission capability, but they can still move. The protein can still move. So the protein that in those dark region keeps moving, and then some of them moving out this region. And then the protein that has the fluorescence, they can also get in. Okay, so then by monitor this dark region, you will find gradually they recovered. Even they are a little bit dimmer, but they recovered. Okay, so the intensity within this region shows like this picture, this diagram. So firstly, we have the fluorescence. You can see my mouse? Do you see my mouse? Yeah. Good. So you see the anion region. You see initially fluorescence is constant. Then you suddenly use very strong laser, bleach it, then boom, the fluorescence goes down to very low level. And then you remove the laser, and then let the cell just recover. Let those molecules just diffuse. Then we will gradually see the fluorescence going up. Okay, they follow this exponential curve. Okay, this curve, and then reaches the steady state. Okay, so recovery. Now, by measuring these curves, the half-time of this curve, okay, then we will know how fast those particles can move in. The fluorescent particle move in, and the darker molecules move out. We can get a qualitative understanding how they move in, move out. That information contains the diffusion coefficient. Okay, if they are faster, they move very faster, then this delta T is very brief. If they move very slowly, like huge molecule, they move very slowly, then you take a much longer time to get the recovery. Right? So this is actually a very quick way to measure the targeted protein, how they are, what's their diffusivity. Okay, and in fact, this measure is very flexible. You can measure the diffusion coefficient of the protein in different regions of the cell. Sometimes they're different. The region close to the nuclear and the region close to the focal adhesion, the laminar podium, the cell migration front, their diffusivity of the membrane protein is different. Okay, so this is actually a very quick way, and you can choose the bleach area to be small, to be non-invasive. Then you can quickly find out, pinpoint the diffusivity of the whole cell, a different region, whole membrane. Okay, so this is a very useful method. And then after you get this data, there are many theoretical equations available. You can use that to derive the diffusion coefficient capital D based on those data. Okay? So in practical, there are several things we need to bear in mind, that be careful when we do this experiment. So one is how to ensure simulation. So simulation actually is what I mean, that using there are many theoretical equations and a simulation method to derive the diffusion coefficient based on the data here. Okay, but based on different geometry of the cell, because cell change different geometry, and the cell have different boundary conditions, right? Different shapes, different conditions. So in different conditions, we need to make sure the simulation really capture those geometry and those boundary conditions, so that they can reliably give us appropriate parameter, give appropriate results. Okay? What if we bleach the molecule in a very weird pattern? Okay, here we show is elliptical, but sometimes if your laser has some issue, you bleach it in a very different way, very different pattern, very weird triangle shape or different shape, then the recovery of the molecule, the dynamics could be different, right? Because you measure the intensity inside, so the recovery could be different. And also, if the cell also show different shape, different geometry, then the calculation will deserve more careful considerations. Okay, and also then so because in this case, then in those weird cases, the dynamics of the recovery might not fit a simple formula, right? May not fit that. So then we need a really careful calculations. And also how to determine whether we should for this whether we should fit a 1-dimension, 2-dimension, or 3-dimension, or mixed diffusion, because in some cases those cells they have some of the molecule they are sitting on a 3D geometry, or this molecule actually can go out of the membrane, so they have a very complex diffusion. Then we all need to consider those specific cases to make sure we can use this FRAP method. Okay, if the condition is very weird and then makes the FRAP calculation to be very complicated, then we should choose other methods, other approaches. So there's a third method. The third method actually is called a FIP, FCS, fluorescent correlation spectroscopy. This is actually a very useful method many years ago, that people use this to quickly measure single particles diffusion. They can actually be high throughput. They can measure many different particles. Okay, so right now it's 3:54. So our class has to stop here. I can explain this in the next lecture, and then you can also read the homework. In the homework, so I put a very detailed explanation of facts, the mechanism, and the calculation method inside. You can read that, and that will help your homework number 3. Okay. Good, very good. Any other questions? Very good questions. So in the class, I think you all of you should really jump in, ask bold questions, ask a very bold question. That will help all the classmates and help us to clarify things. Okay.